Bismillahirrahmanirrahim. Assalamu alaikum. Dear students, I hope you all are fine and well at your home. I welcome you in Pakistan International School Taif's virtual learning class for biology, and this is lecture number 20 of biology second year. Dear students, we have started chapter number 17 in which we are discussing nervous coordination. Yet we have studied all about the nerves, structure, functions, as well as its types and nerve impulse, how it is generated and how it is conducted throughout the nerve. Now we will see very important and interesting topic also, that is synapse, like how the nerve impulse is transmitted from one nerve to another. So let's get started. Yes, dear students, if you see here the topic synapse, so you can see that synapse is actually, what is this? This is the junction between axon terminal. This is axon terminal of one neuron and the dendrite of other neuron. This is the actually connection from where the information from one neuron to another neuron is transmitted or relayed, means handed over to another neuron. That is called synapse. First we'll see its structure, then we'll see its detailed transmission and function, how does it happens. So if we see, this one is synapse. The axon is having axon terminals at the ends, and these terminals look like this, and at the end they are having a knob-like structure. These are called axon terminals or knobs. So these terminals are not in direct contact with that next neuron or next dendrites of the neuron. So there is a space between these two, this terminal, or this is called synaptic knob, which represents this area. Synaptic knob, or this is called axon terminal. So that is not in direct contact, but it is having a space gap between two neurons, that is called synaptic cleft or synaptic junction. The first, this terminal or this knob, that is called presynaptic neuron, and the second one is called postsynaptic neuron, because this is synaptic cleft. So before that we are having presynaptic neuron, and the second one is postsynaptic neuron. And the space between them, that is called synaptic cleft or synaptic junction. Okay, let's talk about its mechanism. It's actually what happened, and that is the number one step. What happened here when the action potential going on through all this axon, it reaches towards this synaptic knob of axon terminal. There are lots of vesicles formed by the cell of neuron itself. These vesicles or bubble-like structures, these are having inside some chemicals, like some chemicals are present over there. These chemicals are called as neurotransmitters. These neurotransmitters, like neuro means neuron, transmitters means transmit, it transmits the messages of one neuron to another, and these are called signaling molecules between two neurons. So first step is when axial potential arrives to this knob. So what happened? Vesicles fuse with plasma membrane. Dear students, here in these knobs or terminals, the neurons are having calcium gates. So calcium is very much important here. They're having calcium voltage gates when action potential reaches here. So by that action potential or voltage, that those all calcium gates are going to be opened. When they open, so calcium from extracellular fluid enters into this knob, synaptic knob or nerve knob. When this calcium enters into the knob, they stick together, they combine with these vesicles. When they combine with these vesicles start to reach to this post synaptic neuron, like at this cell membrane, neural membrane. And then the next step arises, like when these vesicles fuse with the plasma membrane, you can see here like zoomed diagram. This is synaptic cleft. And when they reach to these vesicles reach towards this post-synaptic, like presynaptic membrane, then the next step is exocytosis. Like by exocytosis, this just removes all these neurotransmitters out into the synaptic cleft. So neurotransmitter is released into synaptic cleft, and then these neurotransmitters bind with receptors which are present on receiving neuron, that is postsynaptic neuron as well. And these are called ion channels, and these are also called chemical receiving channels. Like by the binding of these chemicals which are neurotransmitters, these gates are going to open. You can see there they are closed and here it is open. When this is open, so this will just allow the sodium ions which are present here outside the membrane. So they will move inward, and then we can say that action potential is generated here, and there is the distribution of charges again. More sodium will move inward, so there will be more positive inside and negative outside, and the same happen, like it will generate, like it will conduct, we can say, the action potential, and it will move towards all around the neuron, in the next neuron here, like that, from all the sides. If these terminals are around these dendrites, so these all action potentials are get together here in what we can say axon hillock, and then they move towards the axon, and then again to the next terminal, and then again to the next neuron. So there will be distribution of charges again, and like that this membrane gets depolarized, and then it just transmits the signals forward, and then it will become repolarized. Okay, so what will happen in this, will have a closer look here. The receptors, just when neurotransmitters join with the receptors or fuse with the receptors or attach with that one, so ions, we can say, the moves here. And After just when these ions move inward, there is a depolarization. So what happened? These channels get closed. How they are closed? This neurotransmitter, which is mainly acetylcholine in the form of neurotransmitter, which is present when there are muscle contraction especially, and but most of the processes of the body functions which are related to the neuron, they are taken out by this neurotransmitter, that is acetylcholine, and that is broken down into its into separate compounds, and by the enzyme acetylcholine esterase, that we can say that that is hydrolyzed by acetylcholine esterase, and that is broken down um, into like separate substance and components. Then from here when they are broken down, they move back into the presynaptic, inside the presynaptic membrane and the previous neuron. So what happened then? These again they join here and they form what? The acetylcholine, and these membranes form these vesicles. And with that, this is excitatory post-synaptic potential is like which is produced here, and that is up to the threshold level, which is done in this um, like in the next membrane, in the post-synaptic membrane. There is another, like any type of neurotransmitter can perform the function. If that is adrenaline, that is hydrolyzed by monoamine oxidase. So and that all is happen here in this synaptic process, synaptic transmission process. So this gap junction or synaptic cleft, that is almost 0.2 nm, that is nanometer. So that is very small junction here. So that nerve impulse can conduct more rapidly to the next post-synaptic membrane. So like that, the impulse is transferred, or that action potential is transferred from one neuron to another, and then it reaches to the brain or it reaches to any effector. So dear students, now we will see the classification of transmitters, that these are the signaling molecules in the brain, in the neurons, which help to conduct the nerve impulses. Neurotransmitters are actually of two types. They can be excitatory and they can be inhibitory. Excitatory neurotransmitters, they cause increased membrane permeability to sodium ions, because we know that when sodium ions will move in the cell, in the membrane exolemma, or that can be exolemma because the membrane of axon is called exolemma, or the membrane of neuron. So there can be the membrane can be depolarized, and then we can get the threshold level, and then action potential will be generated. So that triggers the nerve impulses, and that's why they are called excitatory neurotransmitters. They are having different types also. Acetylcholine is the main excitatory neurotransmitter for peripheral nervous system. What is peripheral and what is central nervous system, we'll talk about in the next topics, inshallah. But biogenic amines, which are derived from amino acids like protein in nature, so they are neurotransmitters for nervous system. Which nervous system? Central nervous system. So in biogenic amines, we can have epinephrine, norepinephrine, they work oppositely antagonistically as well, serotonin and dopamine. So these are such type of hormones which are biogenic amines, and they are excitatory in the nature. And the next one is serotonin, very, very important amine. They all act as hormones as well as neurotransmitters. Epinephrine and norepinephrine, they increase the heartbeat rate during stress, and we will see in the chemical coordination all about these hormones as well. Serotonin and dopamine, they just affect our sleep, mood. Mood swings can be changed if they are increased or decreased, and attention and learning process can going on by these type of um, neurotransmitters. If we talk about inhibitory transmitters and neurotransmitters, so these are those transmitters which decrease the membrane permeability of sodium ions. Excitatory increase the membrane permeability, but inhibitory, they inhibitor decrease the neuron permeability to sodium ions. So thus they can change the threshold of the stimulus, they can decrease or increase. So threshold level is almost minus 50 to 55, so they can be increased or decreased, so we cannot get any type of stimulation then. So that lessens the chances of nerve impulse will be transferred to an adjoining neuron. So some of the inhibitory are very, very important is GABA. GABA is actually amino acids, and they are called gamma aminobutyric acid. Gamma aminobutyric acid, and the other one is glycine. So the GABA is the fastest inhibitory transmitter, and glycine as well. GABA actually regulates the anxiety and tension, as well as glycine, like um, glycine, it regulates the immune system to limit unhealthy inflammation as well. So they act positively, but they inhibit the process. And endorphins are peptide neurotransmitters that functions for both as hormones and neurotransmitters, which are released and produced in the body, and they decrease the perception of pain in the body. So let's take a look on these neurotransmitters. So if you see, there are different types of neurotransmitters. We have seen adrenaline or non-adrenaline. They can also say epinephrine or non-epinephrine, which we have discussed. So they act for fight and flight response in our body, and they increase the heartbeat in the stress condition. Then we have seen that is dopamine and serotonin. These are like excitatory, um, all these, and glutamate is also excitatory neurotransmitters, and they just act for the memory and conceptions, and they change the mood and pleasure as well. If we see inhibitory, so they are GABA, which regulate our anxiety and then give the calming effect, and endorphins, they give us euphoria, like presence of well-being and feeling good, and they are produced when we do exercise. So our brain cells produce endorphins to make us feel good. So let's take a review of synapse as well. You can see here, this is a presynaptic membrane, and then we are having postsynaptic membrane. So these are calcium channels by which calcium from extracellular fluid just enters into this vesicle, synaptic um, this synaptic knob, and then synaptic vesicles, which are formed by these cells. They are having neurotransmitters in them, and by entering of calcium ions, they just fuse with the walls. And when they fuse with the walls and they do exocytosis, and by that they are just gonna reach to the receptors which are present here out into the post synaptic neuron, and then they fuse over there, and these gates open, and then uh, sodium enters into the cells, and there is depolarization, and the neuron is transmitted to the nerve impulse is transmitted to the next neuron. So you can see this is axon terminal, this one is synaptic cleft, and this is dendrite spine, dendritic spine, like the one dendrite portion of the whole neuron. So by this all synaptic transmission is done, and we can feel the emotions, we can feel the pain, pressure, and etc., because these signals are transferred by this synaptic transmission as well. So dear students, by this we have completed our like synaptic transmission and classification of neurotransmitter. Inshallah, we will see you in the next lecture.